Tēnā koutou, tēnā koutou, tēnā koutou katoa. Welcome to our new version of the SOLA seminars. We have gone public on YouTube, and we apologize again for the technical issues we had last week. We were very happy that Shachi could be with us again this week. So for the question and answer part, we will do it at the end of Shachi's seminar. There's a box on the right-hand side where you can leave your comments and your questions for Shachi. You're free to write those questions during the seminar or after Shachi is done with her presentation. Shachi is a PhD student in the School of Landscape Architecture at Lincoln University. She started last year and she just had her proposal submitted a few weeks ago, and she successfully did a great job. Right now, I'm going to leave the rest of the presentation for Shachi. So Shachi, if you can present yourself and then start your presentation. Thank you.
Sure. Hello, everyone. I'm Shachi, a PhD student at SOLA. And today I will be presenting my thesis proposal. My thesis proposal, which is titled Landscape Design Considerations for Diverse Plant Community Structures that Optimize Carbon Storage Potential of Urban Wetlands in Christchurch. I'm grateful to Dr. Gillian Lawson and Dr. Nada Toyer, who are my supervisors for this research project and who have shown immense encouragement and support at every stage of this project. I would also like to acknowledge the support of Dr. Anthony Shadbolt, who has very kindly agreed to be the advisor for this project. Mr. Andrew Crossland will be supporting this project with his ornithology expertise. In today's presentation, I will be discussing about the research background, the scientific knowledge, and the processes that make terrestrial wetlands effective carbon sinks. This will be followed by a discussion on the research strategies and methods that would be methodically carried out in this project to determine carbon storage capacity of wetland ecosystems. I will be concluding with a note on the contribution of this project on the existing landscape architectural knowledge and wetland science. My project is about urban biodiversity and the ways through which it can be protected and enhanced in cities. It is inspired by United Nations Sustainable Development Goal on climate action, and it looks at ways through which our profession of landscape architecture could contribute towards reducing excess atmospheric carbon from environments. Amongst various ecosystems, this project will focus on urban wetlands, as these are complex ecosystems that are transitional zones between terrestrial and aquatic ecosystems. Wetlands sit at the intersection of three Sustainable Development Goals that are climate action, which is Goal 13, life below water, which is Goal 14, and life on land, which is Goal 15. Wetlands are closely connected to climate change as they are both sinks and sources for atmospheric carbon. This makes carbon calculations a bit tricky for them. Various parameters involved in the design of wetlands that might help with carbon capture, I will be focusing on design considerations of plant community structures and their contribution in optimizing carbon storage potential of wetlands specific to Christchurch only. I will be discussing the background for this project and how it came to be before getting into the research methods and other logistics involved with the project. We have been witnessing global climate change since last few decades, and it is about to reach a tipping point this century. The accelerated increase of greenhouse gas emissions in the atmospheres is due to human activities. There are 6 major greenhouse gas emissions on Earth at the moment, and they are carbon dioxide, methane, nitrous oxide, hydrofluoro and perfluorocarbons, sulfur hexafluoride, and nitrogen trifluoride. Amongst these, carbon dioxide is considered to be a major emitter in the atmosphere. Cities play an important role in these emissions as they emit large amounts of carbon dioxide due to transportation, energy consumption, and land conversion from natural systems to built environments. Atmospheric levels of carbon dioxide started to rise in early 1800s. The first recording of atmospheric carbon dioxide was done during pre-industrial time, where it was recorded to be around 280 parts per million. During the second phase of the Industrial Revolution that began after 1950s, this value started increasing steadily, with the current measure being approximately 417 parts per million. In 2014, we crossed the 400 parts per million mark that was a symbolic target serving as a clear red line into danger zone for climate change. With the current rate of growth in carbon dioxide, which is approximately 2.7 parts per million per year, levels will hit 550 ppm within the next 50 years, putting us on track to cross the global average temperature of more than 2 degrees Celsius, a level that climate scientists say would cause sessions of extreme weather and sea level rise that would endanger life on this planet. Studies show that this Studies show that for this value to be achieved, annual carbon dioxide emissions need to be reduced by 75% worldwide. Many countries committed to work collectively to reduce their emissions within the next decade. New Zealand, which is one of the major emitters, made a reduction pledge of 30% by 2030 and in becoming carbon neutral by 2050. In order to achieve a reduction value of 75% worldwide, we need both reduction and removal measures in place. We are familiar with reduction measures that look at mitigating carbon dioxide levels in atmosphere by reducing emissions through lifestyle changes or with the use of no carbon or low carbon fuel resources or by enhancing energy use with minimal losses. Removal methods, on the other hand, look at sequestering atmospheric carbon dioxide into either terrestrial or aquatic reservoirs with long residence time and secure storage. New Zealand introduced a zero carbon bill in May 2019 that aims to reduce emissions to net zero by 2050. Environment Canterbury is one of the 9 local bodies in New Zealand that declared a climate emergency in 2019 with a commitment to incorporate climate change considerations into City Council's work programs and decision. Christchurch aims to be carbon neutral by 2030. During the 2016-17 financial year reporting period, Christchurch reported 2.9 million tons of carbon dioxide equivalent gross emissions, which equates to approximately 7.8 tons of carbon dioxide equivalent per person. This project is in alignment with the Council's vision of making Christchurch a carbon neutral city by 2030. It will be an attempt to capture the dynamics that optimizes carbon storage potential in the landscape architectural design of constructed wetlands and the ecosystem's processes. Sequestering emission involves fixing and storage of atmospheric carbon dioxide into other pools for long term, where it has a minimal chance of leakage back into the atmosphere. Depending on the soil and vegetation structure, there are many routes to carbon sequestration in terrestrial ecosystems, along with its geologic and chemical uptakes by rocks and minerals. Generally, terrestrial sequestration is based on photosynthesis, with fixing and storage of carbon dioxide into the biotic and soil pools of the ecosystems. Such measures have numerous benefits, which include enhancement and provision of ecosystem services, improving the soil quality, ecosystem productivity, water quality, and biodiversity. Natural systems are being studied worldwide for their capacity to store atmospheric carbon. In 1998, German Advisory Council on Global Change estimated carbon storage capacity and geographical areas of different biomes. Their results are presented in this bar chart. In comparison to other biomes, wetlands, as part of freshwater ecosystems, covered a much smaller area but had a relatively high carbon storage in its overall biomass. And compared to other ecosystems such as forests and grasslands, wetlands are considered the most productive ecosystems with a high soil carbon density. They have a fast carbon turnover compared to other ecosystems. Carbon turnover refers to the average rate at which carbon flows through the stock of the ecosystem. For wetlands, it is approximately 20 years, which this is because wetland plant communities have high capacity to fix large quantity of atmospheric carbon dioxide. There is high nutrient availability and in the soil of in wetland soils, and along with low drought stress. The ecosystem overall has a reduced rate of decomposition with a high organic matter input rate. Globally, wetlands store around 35% of terrestrial biological carbon in spite of occupying small surface area. This makes them effective carbon sinks as they have a high carbon density amongst many terrestrial ecosystems. Wetlands are also heterotrophic in nature, which arises due to their wet environmental conditions that cause high carbon loading in their soil and plant biomass. Wetlands play an important role in local and global natural cycles, as they are at the center of the connection between energy, food, and water. They provide a high percentage, nearly 40% of global ecosystem services, which include atmospheric exchange and regulating atmospheric gases, protecting shorelines, sustaining unique indigenous biota, biodegradation of organic compounds, sequestering carbon, and they act as living filters while maintaining water quality and supply. Wetlands are also considered to be richest in terms of biodiversity, as they are known for the abundance of select avian species that are not commonly found elsewhere. The main reason for such abundance and diversity is the availability of food resources produced by the primary productivity of wetland plant species. Research is an attempt to improve the impact of site design projects on the environment. Research efforts will be directed towards understanding the connection between biodiversity, design of vegetation structures and plant communities, along with the ecosystem carbon dynamics for the entire wetland ecosystems. The aim of the project is to investigate the relationship between plant diversity and carbon residency in selected Christchurch wetlands and explore the implications for designing constructed or restored wetlands in the future. The objectives are to understand the relation between plant species and carbon sequestration, to identify key sites of carbon sinks and plant community structures for optimizing carbon storage, and third, to compare plant communities with avian population as an indicator of biodiversity in the selected wetland sites. I will be briefly covering the carbon cycle occurring in wetlands, which would be helpful to describe the methods that I will be using to measure carbon within the ecosystem. The biological cycling of carbon in wetlands starts from vegetation communities where plants convert atmospheric carbon dioxide to carbon-based compounds such as sugars, fats, proteins, enzymes, and hydrocarbon fuel. And this happens through photosynthesis. This starts the cycle of energy capture within the ecosystem. Simultaneously, when plants die, a share of their above-ground biomass returns to the detrital mass of soil, where it decomposes to form particulate organic matter, or it is also called particulate organic carbon. The decomposition of organic matter by aerobic respiration is fairly efficient in terms of energy transfer. When organic carbon dissolves in water, fermentation occurs that forms dissolved organic carbon, and it moves with both surface and groundwaters. The organic matter, which has been accumulating in the deeper layers of the soil, undergoes further decomposition by a group of bacteria called methanogens. This produces methane under extremely reduced conditions, which flows upward through different soil layers and eventually gets released in the atmosphere as methane. A portion of methane produced in the lower reaches of the wetland soils is interrupted in the oxidized soil layer, and it is converted to carbon dioxide. The gaseous end products, which are carbon dioxide and methane, produced during various decomposition processes, are released into the atmosphere. So this makes wetlands both sources and sinks for carbon, atmospheric carbon in the environment. The two important variables that control carbon dioxide and methane production in wetlands are, first, rise in temperatures. Soil organic carbon pool decreases with increasing temperature as decomposition rates double with every 10 degrees Celsius. The continual increase in temperature leads to more organic matter decomposition that subsequently makes wetlands a major source of carbon. And the second being changes in water levels. Many researchers believe that if wetlands are well drained and therefore also well aerated, they will experience less fluxes of carbon dioxide in the event of global warming. Poor drainage will cause soil anaerobic conditions that leads to considerable amount of greenhouse gas emissions from their ecosystem. There are two factors that determine the credibility of a wetland system to act as a potential carbon sink. The first one is the ratio of methane emission to carbon dioxide uptake for the ecosystem, which gives an index of the ecosystem's greenhouse gas, mainly carbon exchange balance with the atmosphere. If the net value of this ratio is high, then the wetland is a source of carbon. And if the net value is low, then the wetland is considered a potential carbon sink. Another tool is global warming potential, which is used to measure relative potential of greenhouse gases to absorb infrared radiation in the atmosphere. Initially, newly constructed wetlands have high methane emissions, which causes them to have a high global warming potential. Through a dynamic modeling of carbon flux, scientific studies conclude that within a 300-year period, methane emissions become unimportant compared to carbon sequestration in wetlands. Biodiversity plays an important role in helping wetlands become effective carbon sinks. Studies on terrestrial ecosystems have shown that plant diversity helps increase ecosystem productivity, as ecosystems with diverse species richness have a better uptake and accumulation of carbon and enhanced nutrient availability than ecosystems with less diversity in its structure and composition. This happens because plant species, which are the main mediators in the whole carbon sequestration process, they have different capacities to capture, store, and release carbon from their biomass. And it is their collective functional characteristics that drives the carbon sequestration processes in ecosystems productively. In this project, plant diversity will be captured via following parameters. First being the vegetation structure occurring in each wetland, the plant cover, which would help establish species dominance on site, the plant biomass, which would help in understanding the weight of each individual species and the community at large, and the plant diversity and species composition, which would indicate the relative abundance of each species within a given site. Birds are prominent in wetland ecosystems because of its resultant resources, and they are thus considered as one of the visible indicators of the overall productivity of such biotic systems. This project will study abundance and diversity of avian population in selected wetlands and recorded bird species richness data to determine productivity regimes in Christchurch wetlands and assess their habitat quality. The gaps which this research hopes to address are, first, there is a need to understand wetlands at local scales that could resolve the uncertainties surrounding their carbon retention capacity. More information is needed from different wetland types belonging to different regions in the world and their ability to function as both sinks and sources of greenhouse gas emissions. By studying different wetland types in Christchurch, this project intends to establish the role and ability of local wetland plant communities that are part of Christchurch that help in improving the process of carbon storage in its biomass. Wetlands are complex as they are transitional zones between both water and land. There is a lack of systematic models and studies that inform about their carbon sequestration rate and storage dynamics. This project will begin by understanding the natural processes that lead to carbon accumulation in wetlands. It will study the influence of plant diversity, important to ecological landscape design, in enhancing the metabolism of these processes that makes them effective carbon reserves in as short a period of time as possible. Scientific data on wetlands to support the design process is scattered across different resources and forms, which makes it difficult to collect them and comprehend the information concerning their creation or management. The project will analyze wetland plant community structures and create a reference list or a reference index for their carbon sequestration potential and plant productivity, which will support landscape architects and designers in making effective decisions for designing wetlands that will function as carbon sinks. The premise of this project is that a wetland ecosystem with more plant diversity has a higher capacity of long-term carbon storage than a less diverse ecosystem. The project will investigate how, with an increase in plant species diversity, the net primary production of individual species is enhanced, which causes a higher resource and nutrient uptake into the ecosystem and increases the efficiency of its resource utilization. The research questions for this project are: how do plant species influence net ecosystem productivity and carbon storage capacity of wetland ecosystems in Christchurch? Second, how could structural diversity of plant communities optimize carbon storage? And third, how do patterns of avian population relate to plant species and structural diversity of selected wetland sites? This project will use a combination of both deductive and inductive research strategies. Deductive strategy will be used for data collection and analysis, and an inductive strategy will be used to substantiate implications of research findings on design. Deductive approach is often associated with scientific investigation, in which the research starts with the theory, which is tested with data for implication, and it moves from a more general to a specific level, as seen in this diagram. The inductive approach involves working with raw data and identifying patterns in the data that helps to derive themes and concepts. The research moves from specific observations to broad generalizations, as seen in this figure. This project will use a deductive research strategy to identify a relation between plant diversity and carbon storage potential of wetland ecosystems and plant diversity and distribution of avian population in wetland ecosystems. Inductive strategy would be used to relate research findings with the design of wetland ecosystems in Christchurch. Inductive strategy will draw on generalizations from the results and will link them to the design of wetland plant communities that enhance carbon sequestration rate, carbon flux, and carbon storage potential of its ecosystem. The data collection for this project is divided into 4 phases, with each of them being linked with the research questions and objectives. The results of each stage will influence the methods for the successive stage. Phase 1 is about site selection. In this, at least 3 wetland sites will be selected in Christchurch for diversity and carbon calculations. Phase 2 is about plant diversity measurements. That will be done through vegetation surveys on the selected sites in Christchurch. Attributes that help with calculations for plant diversity, such as vegetation structure, cover, density, and frequency, would be recorded and mapped at this stage of the project. Phase 3 is about biomass, carbon, and net ecosystem productivity calculations in selected wetlands that would be done using allometric equations on data derived from the second phase of the project. Phase 4 is about bird species data in all of the selected wetlands for inferring and cross-referencing the productivity regimes of these wetlands. The study area for this project will be Christchurch, as Christchurch is known to be a biodiversity hotspot with many ecological regions of national importance. As per the Council's report, it is known as the wetland bird capital of New Zealand, as at least 100,000 coastal and wetland birds occur here during its peak season. Christchurch City Council is also involved in revitalizing wetlands in order to improve the urban ecosystem services in the city. They are engaged in construction and restoration of large natural wetlands in the upper stream catchments of Avon, avon, Styx, and Heathcote rivers to detain and store excess floodwaters within the catchment. Among the many types of wetlands in Christchurch, the project's data collection and fieldwork will be carried out in at least 3 wetlands, which at this stage are expected to be Styx Mill Reserve, Travis Wetland, and Avon Heathcote Estuary. These 3 wetlands are amongst the oldest wetlands in Christchurch. The reason why these 3 have been specifically identified at this stage is, one, being that they are amongst the oldest wetlands in Christchurch. They also have a well-formed vegetation structure, which would help to identify wetland plant communities, their carbon sequestration rate and storage capacity, along with habitat structure of bird population. They also have a high wildlife value, and conservation efforts are in place to restore native species and improve botanical and bird and invertebrate values on site. Vegetation, within phase 2, the vegetation survey will be done on selected sites, on the 3 selected sites, to record parameters such as plant identification and vegetation structure, plant cover, plant density, frequency, and diversity, along with species composition. They together will help in measuring plant diversity and organic matter biomass for the entire wetland ecosystem. For this, I will be using a transect quadrate method, in which transects will be established throughout the wetland so that each major vegetation zone and habitats are represented. A single transect will be divided into defined frequency intervals, with a minimum distance of 50 meters between each of the intervals, and it will extend through the study area in order to adequately characterize wetland vegetation. The area between each interval will be termed as a quadrate, and species occurring within the quadrate will be observed, photographed, and recorded using different survey methods, which I'll be discussing shortly. The quadrate between each interval will cover at least 2,000 square meters, and within each quadrate, canopy, mid-story, and ground cover taxa will be recorded, along with the water depth and % cover of bare ground on site. The first parameter that I will be looking at each site would be to identify plant species and record existing vegetation structure within the marked quadrate. This will be done with the help of cross-sections that will be set out within each quadrate to mark and identify plant species occurring within that area. Sectional elevation drawings will be prepared at site to capture the distinct layers of vegetation that would help in understanding the existing habitat structure of plant communities in the selected wetland sites. The second parameter that would be observed at site would be plant cover, which provides important information related to species dominance, the habitat quality, and ecological processes such as energy capture occurring within the ecosystem. Plant cover is usually defined as the vertical projection of vegetation on the ground. It gives an approximation of the area over which a plant exerts its influence on other parts of the ecosystem. Plant cover is often recorded at species level, and because of this, it is relatively easy to measure. This project will use a Daubenmeier cover class method to record plant cover in each of the sites. This is a semi-quantitative approach in which we measure cover ocularly in quadrates by using cover classes. This method works by assigning cover to one of the 6 classes that are, these are cover between a tray, first class 1, which is cover between trays to 5%, or class 2, which is anything greater than class 1 but less than 25%, and class 3, which is anything greater than class 2 but less than 50%, and this continues till class 6, which includes 96 to 100% cover of the quadrate. This method requires a large sample size, which includes multiple transects and quadrates in an area, and it is regarded one of the efficient methods to measure cover on site. Third parameter would be plant density, which would be used to describe the vegetation characteristics of a plant community. Density is defined as the number of individuals in a given unit of area. It is used to describe closeness of plant species to one another, and so it relates to the way individual plants occupy space in a landscape. Density indicates trends and rate of change of species in a landscape, thereby indicating their population stability at a given moment of time. This project will use counting methods approach to measure density on each of the sites. Counting methods are plot-based or quadrant-based methods in which the number of individuals found within the plots are counted to estimate density. The units are reported in terms of square meters, which is number of plants per square meters. The fourth parameter would be plant frequency. Plant frequency focuses on occurrence of species within a quadrant, and it will be used to assess vegetation trends across the 3 wetland sites. Plant frequency reflects the likelihood of encountering a particular plant species in an area. It is calculated as the proportion of the number of plots in which a species occurs relative to the total number of plots that are examined. The frequency calculation is quite straightforward, and it is always reported as a percentage, which is we multiply this proportion by 100. To measure plant frequency at each site, I will be using a nested plot analysis approach, which is an objective and a repeatable method that helps in rapid measurement for frequency. Frequency estimates are directly related to the size and shape of the quadrant, and this index is sensitive to abundance and pattern of plant growth. Plant diversity would be the fifth parameter, which will be calculated to compare variation in species presence, their number, and relative abundance on each of the selected sites. It can be considered over a wide range of scales. In this study, I will be using a quadrant by quadrant approach to measure plant diversity, and I will be comparing diversity amongst the 3 sites using one of the similarity indices. Diversity measurements are calculated of composite measurements between species composition, similarity indices, and diversity indices. Species composition reflects the relative contribution of individual as part of the whole for the entire vegetation. Density or cover data are used to calculate species composition, and it is often reported as a percentage or a proportion. Diversity indices such as the Shannon-Wiener index and Simpson index will be used to determine the relative contribution of different species to the entire plant community. This will help in calculating the relative abundance of species and the occurrence of dominant species in a selected wetland. Similarity indices would be used to make comparisons of number of species shared between each of the 3 sites and the similarity of relative abundance of species found on each site. To calculate this, the Chekhonowaski coefficient and squared Euclidean distance indices would be used. The advantage of using diversity indices is that we are able to assign a single number that reflects the diversity of the site. To summarize, parameters such as vegetation structure, plant cover, plant density will be surveyed and recorded during the field visits to each of the selected wetland sites, and parameters such as frequency and plant diversity will be derived from the vegetation structure, cover, and density that is captured on site. And together, these 5 parameters will lead to calculation of plant biomass for wetland plant communities in Christchurch. Plant biomass is the weight of living plant material contained above and below a unit of ground surface area at a given time. Above and below. It is expressed as weight per area and is commonly expressed as kilograms per hectare or gram per square meters. When carbon is captured by an ecosystem, it gets stored in its biomass, in the biomass of its vegetation. This biomass comprises of the vegetation form existing both above ground and in the root system structures that are present below ground, which together influence the soil organic matter pools, carbon turnover, and the carbon sequestration rate for the entire ecosystem. Once individual plant species are identified from phase 2 of this project, Their plant biomass will be determined through a review of literature. This will be done for both above ground biomass and below ground biomass. This data will be extrapolated to the plant community level and wetland ecosystem scale with the help of measured species richness and foliar cover that have already been that were determined in an earlier phase. And this data will be compared for each plant community and across all communities present in a wetland. Carbon pool of wetland vegetation will be derived from plant biomass using formulas identified from literature review. At this stage, I have identified diverse methods that are stated by these authors, which help in calculating carbon sequestration rate and their storage capacity of plant communities. However, the key variable in most of these methods is plant biomass, and I will be able to recognize the appropriate method for carbon calculations once I have gathered the biomass data from each of the selected wetlands. At the ecosystem scale, the net effect of total quantity of carbon entering and leaving the ecosystem is measured using the net ecosystem productivity, which is also known as NEP. NEP includes fluxes from vegetation, detritus, and mineral soil and is an important descriptor of the overall functioning of the ecosystem. It quantifies carbon accumulation or loss, and because carbon and nutrient cycles are strongly coupled, it allows inferences to be drawn about the loss of essential nutrients from the ecosystem. NEP is calculated as a balance between photosynthesis and ecosystem respiration at ecosystem level, which for this project would be the wetland itself. And it provides a better measure of terrestrial ecosystems' ability to sequester atmospheric carbon dioxide concentration than primary productivity calculations alone. All measurements for carbon calculations will include contributions from above ground vegetation and underground soil processes. To summarize the carbon calculations for this project, I will first be identifying plant biomass of each individual species, which will be followed by biomass calculations for different plant community structures occurring in the wetlands, which would then lead to net ecosystem productivity calculations for the entire wetland ecosystem. NEP calculations will be cross-referenced with the two factors that establish the credibility of wetlands to act as effective carbon sinks, which are the ratio between methane emissions and carbon dioxide uptake and the global warming potential of these greenhouse gases. This would be done to determine whether the three wetlands are acting as carbon sources or sinks. What I hope to establish through such measurements and calculations are the implications for design of plant communities and habitat structures that store excess atmospheric carbon as biomass for longer periods of time for both constructed or restored wetlands in Christchurch. In order to evaluate the habitat quality of the proposed plant communities, bird population and their distribution will be analyzed at the selected sites. Birds are often chosen as indicators of biodiversity and habitat quality in natural ecosystems, as they respond well to the availability of habitat structures. The abundance and distribution of bird species have already been recorded within each of the studies site by Mr. Andrew Crossland, who is an ornithologist with Christchurch City Council. The collected data will be analyzed using statistical packages. A diversity index would be used to measure species diversity, and models will be used to test the effects of research variables on bird species diversity and abundance. This diagram shows the bird flyway diagrams for Christchurch, which have been adapted from a publication from Mr. Andrew itself. This pathway is the Pegasus Bay flyway diagram. This one shows this one is the Ellesmere bird flyway, and this one is the Waimakariri estuary bird flyway. The table summarizes the overall research design for this project. Work scheduled to address each of the research objectives have been identified at this stage. I am expecting that by early next year, I will have some of the preliminary results for each of the research questions for this project. Studies show that natural wetlands take around 100 years to become effective carbon sinks. However, with improved design and management practices for both constructed and restored wetlands, this time period can be reduced. It is the aim of this project to develop spatial design considerations for wetland plant community structures that help in optimizing carbon storage capacity for wetland ecosystems in Christchurch. There are a few limitations to the research. First one being that this research project, which is centered around biodiversity and carbon sequestration in wetlands, is a comparatively new undertaking in the field of landscape architecture and wetland science. There is substantial amount of information about wetland resources and management. However, this information is spread across a variety of resources and different forms, which makes it difficult to obtain reliable and comprehensive information on their current state and management. This is also one of the challenges for this project. New Zealand, wetland inventories are generally prepared at national level. In Christchurch, under the biodiversity strategy, the City Council is identifying and conserving wetlands with much work still underway. Difficulty in gap identification within existing inventories is expected to make data collection for each plant species a challenging task for this project. The recent global scenario of COVID-19 pandemic outbreak led to launching of many restrictions for health and safety reasons in New Zealand. Such restrictions, especially in terms of movement, will make field work related to on-site surveys difficult this year. The larger goal of this project is to determine ways through which landscape architecture as a discipline contributes towards climate action through design of urban ecosystems. Wetland science and its aesthetic application is a complex domain, and so its creation and design requires an interdisciplinary effort. Landscape architects play an important role in designing such natural systems to improve their functional attributes, along with aesthetic and human engagement in the landscape. This project aims to provide effective data whose outcomes will comprehend design indices and considerations that promote enhancement of biodiversity in city-scale projects. The intent of this project is to support landscape architects and planners to determine the net ecosystem productivity capacity of the newly created or restored wetlands in Christchurch. It is to help them take measures that optimize the carbon reserve capacity of wetland ecosystems, thereby transforming them into constructive carbon sinks. Thank you so much for listening patiently. If there are any questions, please feel free to ask them.
Thank you, Shachi. Thank you very much for a very interesting presentation and for being patient with us in postponing your talk for another week. You have shown us a very cumulative perspective on your research project, but it's really important to note that it's a work in progress. And Shachi is 1 year into the process, and there are 2 more years to go with the data collection process and analysis. I'm gonna move on to the questions. So we have a question from Chrissy Williams. For the estuarine wetland, how will you allow for the variability of twice daily inputs, including of carbon, from tidal flows?
Right now, I'm only looking at plant community structures, and there are many factors which influence carbon capture in wetlands, including other parameters from other climatic parameters such as temperature, precipitation, etc. But I would be only focusing on the plant community structures for the entire wetland ecosystem.
Thank you, Shachi. We don't have any more questions from the audience, but maybe we can wait for another minute if anyone else has any more questions. Okay, well, thank you everyone for following the public online event. We will resume next semester with a new list of speakers, and with having access to YouTube, we might be able to expand the audience for the SOLA seminars. Thank you again for joining us, and we look forward to seeing you again next semester. Thank you, Shachi, for your presentation.
Thank you, Nada. Thank you so much.